screen is not on.
Hey, Jonathan. How are you? Okay.
Hi, Fatih.
Yeah, let me start the seminar. Hello, guys. Welcome to today's Zoom talk organized by the Single Cell Immunophenotyping Core. It is my pleasure to introduce our distinguished speaker, Mr. Jonathan Matthews, a master user of our core and accomplished scientist. And he will be graduating as a PhD in bioengineering for PME in June, right? And has an impressive academic background in electrical engineering and computer science from the MIT. And today, he will share his expertise on effectively handling sequencing data, BSL files and all the data in Basespace, and focusing on monitoring Illumina sequencing runs in Basespace and transferring data to RCC and 10x genomics platforms. Please join me in giving a warm welcome to Mr. Jonathan Matthews. And let's dive into the informative seminar together. Thank you. And by the way, also we will record this talk. So soon we will put this talk to YouTube channel, our YouTube channel. Okay, thank you very much.
Great. Thanks so much, Fatih. Yeah, so as Fatih mentioned, I mean, I'm soon to be a PhD. I'm just finishing up my graduate studies. So I have no affiliation with Illumina or 10x Genomics, but I have spent probably like the last year or so working with the single cell sequencing pipeline. And it can sometimes be difficult to get started learning how to work with the sequencing data. So I thought that this might be a useful thing to share. And yeah, hopefully you guys find it useful too. So let's see. So the overall point of this is, right, if you start with a DNA library, so there's a lot of different platforms that can be used to prepare your DNA library. You can work with Illumina library prep kits, or you can use 10x Genomics kits. But what I want to talk about is how you go from your DNA library to all the way to actually being able to analyze single cell data. And importantly, how do you do that efficiently? How do you save time, maybe even save a little bit of money while you're doing that? So this is really the big question mark that hopefully you'll gain some insight into how this works. So to expand on how this works a little bit, of course, when you have your DNA library, you're going to start by adding it into a flow cell. So this is all using Illumina.
RNA minus 80 and the.
Oh, I think that we should probably mute someone. Let's see. Okay, I think we're good. Yeah, so this is all going to be using Illumina for sequencing and 10x Genomics single cell sequencing technology. But a lot of the stuff that I'm going to talk about would be applicable to other platforms too. So you have your DNA library, you're going to load it onto a flow cell. You put this into a sequencer, which does some magic to determine the sequence of DNA that's in your original sample. This information is then going to get streamed to the cloud. So Illumina's cloud platform is called Basespace. Sometimes it's called the sequencing hub. But this is just Illumina's interface for dealing with sequencing data. So you don't actually have to physically be present on the machine. You don't have to use like a USB stick to get your information off. Everything just gets streamed online in the cloud. When you're doing single cell, you then want to use a platform such as 10x Genomics Cell Ranger. And this is going to take all the sequences that your Illumina sequencer measured and figure out how to assign them to individual cells, right? So a lot of the 10x technology is about figuring out how to barcode the sequences that you're going to get, that you're going to read out with Illumina. And the Cell Ranger technology is what's going to figure out how to group these different reads together to assign them to individual cells. So what I'm going to talk about today are these different steps in the pipeline. So sort of understanding what actually happens when the sequencer is transferring data and what kind of data is being sent from the sequencer up to the cloud, because that data is important to understand because you can monitor this in real time. So sequencing sometimes can take 24 hours, maybe even longer for really large read kits. And so it could be useful to understand what's going on here so that you can monitor runs in real time. At the Basespace level, I want to talk about a couple different file formats because these end up being pretty important if you're working with the sequencing data directly, but also just so that you have the vocabulary as you see BCL and FastQ files, just sort of this alphabet soup, sort of having an understanding of what's actually going on. I'll also talk about how you monitor runs and interpret quality. I have some examples of what things look like when they don't go right, when they don't go as planned. Then talk about how you can actually transfer that data from the Basespace platform onto a system that can run Cell Ranger. So Basespace has a lot of really useful apps that are built into it, but I don't think that they yet have Cell Ranger or any 10x tools built into it yet. So you do have to transfer your data from Basespace to a different platform, which then you can use to run Cell Ranger and do your single cell analysis. And in particular, I'm going to talk about my preferred method for this, which is to use the 10x Genomics cloud analysis platform so that I don't have to actually run the exact Cell Ranger commands on a computer by myself. I can use their cloud system, which has a really nice user interface, and they do offer it for free, which is really nice because you don't spend those usage dollars on the resources that we have at UChicago. This is really a nice tool that 10x has provided for its users. So just to go back to what a sequencer is really doing, so just really simply, the sequencer is just taking really high quality images of the flow cell. It's going to have an image for each cycle in the flow cell. This is sequencing by synthesis, so every dot that you see here is a set of fluorescent reversibly terminated nucleotides that's being added to a cluster that corresponds to a single DNA molecule from your original sample. And so by doing this over and over again, all the sophisticated equipment and fluidics inside of the sequencer do this over and over again so that you get all these images for each cycle, and the color is going to correspond to what nucleotide's been added on. But this is really, really high density information. This is just a small window into a flow cell. There's a lot of stuff that's going on. So the sequencer doesn't actually store these images in this raw format and uploading those to the cloud, because that's just a little bit excessive. It's not really what's needed. Instead, what's done is it uses this BCL file format. So BCL file format, BCL is, I think, just base call. And instead of storing a whole image of each cycle, for each cycle, it is a list of what base is in each cluster and the quality score for each cluster, right? And so you're going to have one file for each of these cycles. So for cycle 1, instead of that 2D image of all of the colors at each cluster, it's going to store just a list of what base was considered to be the most likely base at each cluster. And this is a lot more efficient. So what are these quality scores? This is an important thing to see. This is based off of the Fred quality score metric. And this is a logarithmic relationship so that you can get, because single cell sequencing, or sorry, sequencers are actually very accurate. It doesn't really make sense necessarily to say, that a quality score of 1 is like 1% accuracy, 2 is 2%. These sequencers are really accurate, and so it becomes more important to say, is this 99% accurate versus 99.9% accurate? So that's why this is all logarithmic. So you can see the relationship between the accuracy and what the Fred quality score is here. So if you a particular base has a Fred quality score of, say, 30, that means that it's a 99.9% chance that that base is correct. So you can see these files on Basespace, and I'm going to do the risky thing here of actually trying to do some stuff in real time, just showing you some stuff on the site. I think that that's important just because sometimes things don't go as smoothly or as fast as you want them to go, so it's good to always see what they look like in real time. So this is the Basespace website. This is where everything is going to get uploaded to from your sequencer, and this is happening in real time. Stuff is going to be uploaded after every cycle is computed on the sequencer. That single BCL file is going to be streamed and uploaded to Basespace. So here's a list of runs from our lab at UChicago. This is the TI lab at UChicago. You can see we have done a lot of sequencing over the last couple months. I'll just look at an example one. So this is one that I did a couple months ago. And this is sort of what I mean. I want you to appreciate the timing for how these things work so you don't get spooked if you're like, oh, this is taking too long to load. This is just, there's really lots of data behind the scenes here, so it sometimes takes a while. So this is like the run front page. You can see some overall statistics. But just to talk about those BCL files, these are real things. If you go to Files here, you can drill down and you can look in the data folder, right? These are all files that are generated by the sequencer, and you don't really need to go into the details of these. You don't ever really need to interact with them. But just so that you'd appreciate that this is really what's happening in real time, you can go to Data, Intensities, and you see this Base Calls folder. And you'll see for all the different lanes on the flow cell, we have a whole bunch of BCL files that are generated for each cycle on the flow cell and for different tiles. So this is going to be streaming in real time, but it's not really useful to be looking at this. We want to actually be able to measure those quality scores in real time. And so Basespace has this really useful panel here, which is charts. And this will be, I'm just going to say it over and over again because it's such a useful thing, is that in real time, this is going to be added onto. So when I was doing this, this was one of my first sequencing runs. I was in a different state when this was running. One of my lab mates actually set this up. And so I was able to just monitor the run on my phone, which is great because I was very worried that it wasn't going to work well. But it ended up working pretty well, and it was nice to just follow along with it. So over here, you can see data by cycle, right? So this is going to be adding in a new data point after every cycle. And there's a lot of information that you can look at here. The one that's probably the most important to look at is the quality score. So percent greater than Q30. This means of all the clusters that are on the flow cell, what percent of them have a quality score that's greater than that Fred score of 30. So you can see that this is what it looks like when the run is completed, right? And this is expected where it's going to slightly go down towards the end of a read. But overall, this looks pretty good, right? We're getting over 90% quality scores for pretty much all of these bases, which is great. There's some other useful things that you could look at. So for example, you could look at the percentage of base calls at each one. So in most libraries, your data is going to be sitting at around 25%. And that makes sense because if your DNA library is sufficiently diverse, you should expect that at each cycle, there's a roughly even share of each one. But then you'll see this region here, which is where most of them go up to 100, right? And this would correspond to an index sequence. Right, this is where pretty much all of the clusters on the flow cell have the exact same nucleotide sequence. And so this is how you can sort of validate or things where you would expect. If you're working with barcode sequences, such as with 10x technologies, with cell surface profiling, cell surface protein profiling, you'll be able to see different regions that correspond to barcodes that would be more or less abundant. Another useful tool down here, which I really like, is the flow cell chart. And this could be useful for just checking that your flow cell is working well. And I'll show you one more thing that's really nice about it. So the flow cell, right, this is where all of the clusters, all of the oligonucleotides are being adhered onto, and this is what's being imaged by the sequencer. But you can look, and there's tons of information on here as well. We can look at those Q30 scores, and instead of just looking at it by cycle, so again, why don't I go back and switch to the Q30 score by cycle. And you'll see that there is some variance at each cycle. And so you might wonder, well, is that due to maybe there's some less performance on a particular part of the flow cell? So you could look at that here, right? And you can see what are the quality scores for the different oligonucleotides across the flow cell. This looks pretty good at this point. Everything is the colors are just scaled to the min and max. But you can see that the minimum here, this dark blue, is 95%, which is already pretty good. But I'll show you some examples where things go a little bit awry. Something else that's nice that I like to look at is you go down here. Again, there's so much information that you can look at. Something that I find useful is to look at percent aligned. And what this is talking about is how much of the clusters that are red are aligned to the phi-X genome. And this is important because if you're dealing with a library that has low nucleotide diversity, you can add in this additional sequence, phi-X, which is, I think, this bacteriophage or some kind of virus. But this is a very well categorized genome, and this is something that will just increase the diversity of your sequencing library, which will give you much more reliable measurements for each cluster. The machine uses this to sort of calibrate all the intensities for things. But so this is a nice way to validate that, okay, when I was preparing this DNA library, I wanted it to be around 1% phi-X. And I can see how accurate that was. Did I pipette well? In this case, did my lab mate pipette well? And she absolutely did. Got around 1%, 1.24%, 1.36% phi-X for all these different tiles. So that's really nice. I want to show you what things look like when they don't go to plan. So what would have happened if I didn't put in an appropriate amount of phi-X for a very low diversity library? So here's a different run. I'm going to go back to charts here. Again, I'll look at the quality scores just because that's a nice thing to look at first. And you'll see, wow, this is all over the place. What is happening here? So it looks pretty good for these first couple bases, but now we're getting quality scores that are all over the place, really, really low, down at 40%, 25%. What's happening here? So something else that would be useful is to look at the bases and see what bases does this correspond to. And you'll see that if you match this up, that a lot of those bases correspond to regions of low nucleotide diversity. And so this is highlighting the importance of using phi-X in your sample. You might see problems like this if you don't have sufficient phi-X. You can then go and look at, again here, the percent aligned. And you'll see that this is low. It's still around like 1%, but that previous example was for a cDNA library. This example is for a cell surface protein library. And so for this, you're going to want to use a lot more phi-X, like 10%, maybe even up to 40% phi-X, to be able to compensate for the nucleotide diversity. But this is the sort of thing that you would see when things aren't really going as expected. And you can see that reflected again in real time during sequencing. You'll be able to see that stream through. I think I have one more example. Yeah, this is where you might have a problem with your flow cell. And this can be a little bit more difficult to track down, but I can just show you an example here. So again, I'll look at data by cycle, and I'll look at my quality scores. And it doesn't look as necessarily bad as the last one, but we definitely see a big spread on some of these, right? So over here at cycle 28, there's a wide variation in quality scores. Here's another example at cycle 39. It's not great. So it can be hard to tell what's going on here. If we were to go look at the percentage bases, we'll see that this is corresponding to the index region, which is not really a factor here. But for the other regions, it's not really seeming like it's due to nucleotide diversity. Or lack thereof. So instead what you can do is you can look at the flow cell chart. We'll look at quality scores greater than 30. And let's look at, like, for example, that cycle 28, I think it was. And yeah, you'll see that there's a big strip here, which corresponds to lower quality scores across this tile. So this might have been an issue with the way that I loaded the flow cell. It might have just been an issue with the flow cell itself. But this kind of gives you an indication of what actually went wrong here. So a lot of these, right, this strip here has over 90% Q30. But for these runs, it seems pretty spatially defined, where these tiles are much lower, 58%, 40%, and so on. And you can see this in different cycles across the whole thing. So that's a way that it might give you an indication of what's going on there. Yeah, I'll get to the last one, the other example in a minute. Okay, so that's just sort of a little walkthrough of Basespace. And really, this is all just, again, stuff that you can see in real time that's streaming. The BCL file format is really nice because the sequencer can just stream one BCL file up to the cloud after each cycle of sequencing. But at the end of the whole sequencing run, we don't really necessarily care about the distribution of bases at each cycle. Having files that correspond to the base at each cycle is not really that useful to work with. And so there might be a better format to work with that will let us work with entire reads, group things together by reads. And that's the FastQ file format. That's really the power of this file format. So instead of the BCL file format, which was one file per cycle, and each file just has a list of bases at each position and their quality scores, a FastQ file, there's one file that's generated per sample, right? So if you have multiple tissue samples that you're sequencing from and you have those indexed differently, you'll get a different FastQ file for each one. And what this does is it's going to combine multiple BCL files together, the information from multiple BCL files, and it'll, instead of giving you a list of cluster bases, it'll give you a list of cluster sequences. So here's an example of what a FastQ file looks like. A lot of times you don't have to deal with these files directly, but in some cases, especially if you're in technology development, like working with new methods for sequencing, like some of the stuff that we do in the TI lab, it's important to understand how these work. And I think it's just always nice to look under the hood and see how these things, what these things look like. So here's an example of a FastQ file. So you'll have some entries for each cluster. And again, these clusters basically correspond to one nucleotide in your original sample. And so here's listed the sequence of nucleotides for that sample in orange. And down here are the quality scores. Now, looking at this, you might not know what these correspond to. Is this like fair, not so bad? I don't know what these will correspond to, what these letters correspond to. So it's kind of interesting to see how these are encoded. So this is done with ASCII encoding. And so here's an example of an ASCII table. And the idea is that every character has a number that's corresponding to it. So for example, the letter J corresponds to the number 74. The letter K corresponds to the number 75. And you'll notice that the very first number that corresponds to a character that you can actually see on your screen, all these are just sort of control characters or spaces, things that you don't actually see visibly typed. The first one is down here, number 33, which has an exclamation point. So 33 corresponds to an exclamation point. So that means that if we are dealing with a base that has a Phred quality score of, let's say 0, let's say it's terrible, we're going to assign that to be the ASCII code 33, because now we can represent it in our FastQ file as an exclamation point. As another example, if we have a Phred quality score of 10, right, that's going to be 10 more than that initial, first text representable number, 33. It's going to be 43, which is a plus sign. And so if you go back and you look at this file here, right, you see that there's F's and H's, J, I. So what quality scores do these correspond to? What accuracy is this representing? So an F corresponds to the ASCII number 70. So the Phred quality score, we're going to subtract 33 from that. That gives us a Phred score of 37 for this base, which is 99.998% accuracy. So that's just sort of how this stuff is encoded. And when you're looking at FastQ files, that's just sort of what this line looks like. Again, it's just sort of adding to the alphabet soup of dealing with all these different file formats. But I think that this is something useful to understand if you're working with this kind of data. Okay, so Basespace does all of this work for us of turning the BCL files into FastQ files. I think that 10x Genomics also has some software that wraps around the Basespace technology. But fundamentally what's happening is this BCL Convert software is turning all of the BCL files that have been gained during sequencing and converting them into FastQ files. But it actually does a lot more than just converting. There's a lot more that's happening with BCL Convert. So for one, right, each sequencing cluster is going to correspond to multiple reads, right? So if you have paired end reading, if you have dual index reading, you're going to get a lot of different groups of sequences of nucleotides that you want to handle separately. So you can generate, the BCL Convert software will generate one FastQ file per read. Again, this is per read and per sample. So for example, if you're doing paired end sequencing, we're going to have one FastQ file that corresponds to read 1, one FastQ file that corresponds to read 2. The other thing that's really nice that it does is it does sample demultiplexing, right? So if you have, again from this last image, if you have, let's say, tissue DNA libraries from different tissue types, and you've prepared them to have slightly different indexes, right, with the sequencing adapters, the BCL Convert software is going to automatically strip all that information and group reads together based on shared index. And it does that according to the sample sheet that you give it. Right? So that's kind of the purpose of giving it the sample sheet when you're doing a sequencing run, is so that the BCL Convert software knows what to call each index that it sees. This is something that you can also see on Basespace. So let me just go back to Basespace. It's behind this menu bar here. So you can see it in Basespace. If you go to, I think it's under biosamples, or sorry, you go to the project that you've created, and you can see your FastQ files that are generated for that project. And so, yeah, so here's a FastQ file that we, if you want, you could select this, and you could go ahead and download that FastQ data set right here. There's one more thing that I wanted to show at this point. Oh yeah, so what happens if you mess up your sample sheet? What happens if you accidentally enter in the index wrong, or there's a lot of other stuff that goes on in the sample sheet. The BCL Convert software sometimes gives the indexes a little bit of wiggle room. So if there's an error in one of the index spaces, it can still match that read to the correct sample. What happens if you set up your sample sheet incorrectly? So you can re-queue the analysis with a new sample sheet. There's slightly different ways that you do this depending on which sequencer you use. So if you're using the MiSeq or NextSeq 500 or 550, It's a really straightforward way to do this. So I think it was this first one that I showed you that was done with an XSeq 550. And if you want to requeue this, you go to requeue sample sheet, right? So this is just the run page. You click on the hourglass requeue sample sheet. And this is really nice because you can load up the original here, you can make any edits that you want to make, and you can requeue the analysis, and it will run BCL Convert again and do that sample demultiplexing with your new settings. It's a little bit different for working with the XSeq 2000. So for this, we actually have to upload the new sample sheet to a different project and then run the BCL Convert app ourselves. And I'm going to show you what that looks like, if I can remember how this works. So let's say that, for example, that was my last one here. So this is a run that was done with the XSeq 2000. And let's say that I accidentally used the wrong sample sheet, which I actually think for this I did use the wrong sample sheet at first. So how can I requeue this? Well, what you would do is, again, I just want to show you like how you get to all this stuff. So you can run the BCL Convert software. So you can search for BCL Convert. And it's an app on the Basespace Sequence Hub. You can pick it right here. And then you launch the application. I'm not really sure why this is so different for the XSeq 2000, but this is what you have to do. You select the run that you're interested in working with. So if I scroll down, I think it was this one that we were working with. Select that. And then you override the sample sheet. All right, so here I would select the correct sample sheet that I've uploaded. And I'll show you how you upload the corrected sample sheet. To do this, you need to actually create a new project. And I'll name it fixed sample sheet. And then we can upload a file. I think that this should probably be simplified if anyone from Basespace is watching. So you can go here, you click on miscellaneous, and then you can go ahead and upload your corrected sample sheet. So scrolling through, I think that this was the corrected sample sheet that I made on my computer for that run. So I'll upload it here, finish upload, this is going to upload to the project. And if I go back to that BCL Convert software where you select the sample sheet, I can select this new file, and that will requeue the analysis. So a little bit convoluted for the XSeq 2000, but occasionally you need to do that. And if you do want to just have like a walkthrough for how to do this, Illumina has a nice article that walks you through it. Okay, so let me continue here. All right, so what happens once we have Basespace having done this BCL Convert software, our FastQ files are ready, everything has been demultiplexed correctly. We want to now run these through Cell Ranger, through the 10x Genomics software platform. So where does this actually happen? You could just download this onto your laptop. This is a totally viable way to do this. You can go on a Basespace and just select the FastQ files and just download them over the internet to your laptop, and then you could run the Cell Ranger software on your laptop. This is pretty slow for downloading, especially if you're maybe working on the go a little bit or working remotely, as people sometimes do with sequencing. So there is a much faster way to do this. You're also going to be running Cell Ranger on your laptop, which, unless you have a very powerful computer, that might run pretty slowly too. So how can you save time and maybe even save money in this process? So we can use the resources at UChicago, the Research Computing Center. They have a really, really fast internet connection, and so they can download FastQ files directly from Basespace really, really quickly. What's also nice is that we can just upload this information to the 10x Genomics cloud. And 10x Genomics has this cloud tool, which I'll show you in a minute. It has a really nice user interface for configuring Cell Ranger and visualizing some of the results. And this is my preferred way to do it. You could also run the Cell Ranger command line tool directly on the RCC cluster, which is totally a fine way to do it. I prefer this way. We don't have to spend the extra usage dollars on RCC. And then additionally, I really like the user interface that 10x has made for it. So yeah, let me again take the risk of walking you guys through this in real time. So you have to go to this nasty command line window, but it actually is not as complicated as it seems. I'm going to make the font slightly larger so that everybody can see it. Okay. So clear this window. So you can log into, you know what, actually, let me just sign in directly so that you can see how to connect to this. So if you're on Windows, my preferred tool is called PuTTY. This is how you can connect to RCC on Windows. You can do this through the terminal on Mac or on Linux. There's terminals too. But this is the way that I like to do it on Windows. You can load in your Midway settings. So this is the connections to Midway, which is one of the RCC clusters. And there's plenty of documentation that UChicago has for how to do this. Connect, type in your password. Of course, you have dual-factor authentication. Try it set to my watch. Okay, and so now we're logged in. And now I'm going to once again make this font larger. So just so that you're all caught up with how I get to where I am. Okay, so this is what it looks like when you've logged into Midway. You can type ls to see what's in your current directory list. So these are the files that I currently have here. This is because I've already downloaded some of the stuff that I'm about to show you. So I'm just going to remove everything I've already had here. So we're going to do everything in real time. Okay, so the first thing that we want to do, if I go back to my slides here, we're going to use the Basespace command line tool, right? And this is what lets you download stuff from Basespace directly to a platform like RCC, which has you just interacting with the command line. If you want to see a reference to this, you can always just Google Basespace CLI, but I have a link here which you could also go to. And there's nice documentation for how to do this. It can be a little overwhelming if you're not familiar with working with the command line. But it ends up being really simple, right? There's just a couple commands that you have to run. So this first command, I'm just straight up going to copy and paste into that command line window. And this is going to download the Basespace command line tool. So I'm just going to paste it, run it. I have to make the bin directory. mkdir make directory bin. And there we go. So just like that, it's downloaded the Basespace tool. If you try to now run bs, which is the command for running Basespace, it'll say that permission's denied. And if you go back to the instructions, that's why they had this additional command here, which you have to run to make sure that this new file that you've downloaded has the right permissions. But you can, again, just copy and paste this. And now when we run bs, it has all of the commands that we can run here. There's a lot of information, a lot that you can do with this, but it turns out that it corresponds pretty well with just using the Basespace website. So the most important thing that you're probably going to want to do, let's clear this window, is bs list projects. So this is going to list all the different projects that you can just see from the Basespace website. And here's the one that I was interested in before. And so what we want to do is download all of the FastQ files from this project onto RCC, onto this cluster. And this is going to be a pretty big file. The FastQ files go up into the gigabytes. And so the way that it works on RCC is that they give you this really nice scratch space, which is just a ton of storage that you can temporarily use, which is perfect for this case where we just want to download the FastQ files and then immediately upload them to the 10x cloud platform. And that's nice because then it doesn't eat away at your own RCC data storage. So here's how you download stuff. I think I also have this here. Oh yeah, so list projects, you can sort them if you want. There's a lot of info on the Basespace CLI website for how to work with this tool. But what I'm going to do is download the FastQ files from this project. And actually, I'll walk you through this command rather than just copying and pasting it. So when I listed the projects, it shows me a list of all the project names, but it also shows an ID here, which is going to be important to use. So I'm going to, I'll copy this in just a minute. Just as there's bs list as one of the commands, there's also bs download. And if you're interested in what the commands are, you can always just type -h, and this will list all the options that are there for you. So I'm interested in downloading a project, bs download project. And again, I can do -h to see what the options are for downloading a project. And it gives you a lot of options that you can type in. The important one here is going to be ID, right? So this is where we're going to give it the ID of our project. You could also give it the name if you want. but I'm a programmer. I like working with just the numbers in case there's a duplicate project name. So you can use the ID. This is another thing that'll be important is the extension, right? There's a lot of files, as I showed you guys, that are associated with each project. We want to only download the FastQ files, so we're going to add an option for FastQ. And then the last command that we want to, the last option is the output option, because we want this to be saved on the scratch space area of RCC so that we're not dealing with our quota, our storage quota that RCC gives us. So we can type this directly out. So again, I want to first get the ID of the project. So I'll do bs list projects. And here is the project ID that I want. It's this one. So let's add that down there. And I'm going to do bs download project dash ID equals this. Dash extension equals FastQ. I'm pretty sure that you can just say FastQ, but probably a better way to do it is FastQ.gz, which additionally uses a zipped, a compressed version of the FastQ files rather than working with the uncompressed FastQ files. And the very last thing is -O for output, and we're going to say this in Scratch /midway3/Jono. And I'll start running this. What is your, not sure what's going on here. Let me make sure that I copied it correctly. I'll just do it exactly from this. I think that this is exactly what I did. It might be because I already downloaded this stuff. Yeah, it's because I already downloaded this before this talk. So this took about like maybe 10 or 15 minutes to download. I'll show you what it looks like when it's already there. So here are all the files. So pretend like when I ran that command, it took about like 10 or 15 minutes to download. But these are pretty big files. So I'll do ls-l. And you can see that these are pretty huge files. These are gigabytes of data, which would probably take you a lot longer to download on your own computer. But these are the FastQ files that have been downloaded. So I could go look at individual ones. Again, there's an individual FastQ file that's downloaded for each lane that's on this flow cell if you had multiple samples on this flow cell. And here are the different FastQ files for, this is a 10x run, so we have a FastQ file that's for read 1 and a FastQ file that's for read 2. So all this has been downloaded onto RCC, and there's a message in a chat. Let me see. I'll share the recording. Yeah, everything will be shared. Yeah, so you can download all this stuff directly to RCC, which is pretty quick. And the next step is to upload this to 10x Cloud. So let me go back to my handy reference so that I can do this, try to do this in real time. So just as there's a Basespace command line interface, there's a nice command line interface for 10x Cloud. So before I do that, let me just show you what the 10x Cloud site looks like. You can just go to cloud.10xgenomics.com. You can log in with, I think even just with like a Google account. You don't have to create a dedicated account just for this. And this is what it looks like. So you can have a whole bunch of different projects here, which correspond to each of your individual sequencing runs. So I'll create a new project, and let's say that this is 10x sequencing example. Doing these walkthroughs in real time is always so nerve-wracking. Okay, so we can upload files here directly through the web browser, which again, you can, just as I said before, you could download the FastQ files to your laptop and just upload them right here. But instead, what we could do is we could use the 10x CLI to upload them from RCC, which is a lot faster, and we don't have to deal with storing a 60 gigabyte file on our tiny laptop. So they have nice instructions here for how to set this up. And just as before with the Basespace CLI, we're just pretty much going to copy and paste and follow these instructions. So I'm going to copy this command, go back here. I'm going to go back to my home directory. Let's clear the screen. I'm going to paste this, and this will download the 10x CLI tool right onto my laptop. And I can see it right over here. This is a compressed file, so we're going to need to unzip it. And again, we can just use a command that they have listed on the website, click copy, paste it right here, and now it just uncompressed it. And we can see that it's in its new folder right here. I can then go into that folder. CD is change directory, by the way, if you're unfamiliar with working with command line. And there's this program here, TXG, which is the 10x software tool for how you interface with the 10x cloud. And then, so you scroll down again, and here you go, upload FastQ files. I'll copy this command. And you see that all this looks great. Our project IDs here. The last thing is path to FastQs. So we're going to have to put that in ourselves. And so I'll do /scratch. This is where I'm putting in the path to those files that we just downloaded with the Basespace CLI. Midway 3 /Jono / I think it was student name L001. There we go. And I'll hit run. There we go. You're about to upload 2 files to the following project. 8.73 gigabytes. Uploading is even slower for me on my laptop, so this is another area where things go much faster when you're using RCC. And when you're ready, just press yes, and it will continue. And then it'll start to upload, and it'll notify you when the upload's done. I'm obviously not going to sit through the whole thing here, but I just want you to appreciate like how fast it goes. It's much quicker than what I can do at home. So it's pretty much already done with uploading a 2 gigabyte file onto the cloud. But anyways, I'm going to stop this part, and I'll go back and I'll sort of do what they do on cooking shows and fast forward to what things look like after you upload them. So here's an example of the project where pretend that I've uploaded all those different files. They'll just pop up on this site, and they're listed here. You can select what reads these correspond to. So for example, when I'm doing 10x data, a lot of times your reads are always going to include some gene expression information. Sometimes they're generated with different library prep kits. So if you're doing the high throughput or low throughput versions from 10x, you would use those here. For my application, I was doing stuff with a custom feature barcode library. So you can select that down here. And the reason why it's important to select, specify what type each of these reads are, what FastQ files these are, is because when you do analyses, you want to make sure that the 10x Cell Ranger tool handles these appropriately. So something else that's just I wanted to point out is you can see that these FastQ files, something nice that 10x does is they group it together by read 1 and read 2, which is just good for ensuring that everything is in sync. So what I'll do is I'll just select all these different files, and I can go to create new analysis, and this is pretty much just going to run Cell Ranger for me on the 10x website. So I can specify what pipeline to run. Pretty much always just going to use their most up-to-date version. You'll come up with your name for this analysis, so 10x analysis example. Transcriptome reference, because Cell Ranger with this pipeline, they do transcriptome alignment for you to a reference genome. You can upload a feature reference, because I'm not going to walk through this, but this is if you're working with, as in my case, a custom feature barcode library, you could upload your own CSV here, and 10x has a nice specification for what this looks like. But yeah, once you specify this, you just click run, and then I'm going to fast forward again. You'll see it populated on the analyses page, and it'll take maybe like an hour to run, which is much faster than it would have run on my laptop, and I think might even be faster than it would run on RCC. I'm not sure. But this is a reasonable amount of time for the Cell Ranger tool to run. And what's nice, though, is that now you can see your results on the 10x website in this nice tool. So you see all the files that are output from the analysis. The first thing that it's good to look at is just the results summary. And this will show you this page here of information about how many cells were recovered, reads per cell, and so forth. You can look at gene expression, and this just gives you a very bird's eye view of the different samples in your data set. So by default, the 10x Cell Ranger tool that they have on here will do secondary analysis, so that's like clustering, stuff like that. And so you can just see an overview of it here. Something else that's nice to do, which is probably going to be the last thing that I'll show, is I really like using 10x's Loupe Browser. And that's just a tool that you can use to explore this data a little bit more in detail. So you can just download this file directly. So here's the cloop file. Just be a couple seconds and then it'll be done. I think I already had this downloaded, but I like doing this in real time for completeness and the adrenaline rush, I guess. And I'm pretty sure that that they just have a link for where to download the Loupe Browser. If not, you can always just Google 10x Loupe Browser and just pull up the program. But this is a really nice tool to just explore your data. So this is the Loupe Browser. You can look at t-SNE projection of your data. You can look at a UMAP projection of your data. So in this case, I'm pretty sure that these are some dead cells, which you would detect with high mitochondrial counts. You can do reclustering. And so there's so many options here, so many things to walk through. They're really, you can just play around with it. It's a pretty nice interface. I don't think that you need a lot of expertise to start working with it. But here's where you could do like setting thresholds for the number of UMIs that you care about per cell. So this is where you could get rid of doublets or cell debris, get rid of cells, threshold cells by number of genes expressed. Here's where you can do some mitochondrial filtration. So this shows me all the mitochondrial genes that are in the sample. So normally you want to go down to maybe below 5%. And now you can decide what kind of plots you want it to generate, what kind of secondary analyses. So I'll call these maybe cells, recluster. I don't think it normally takes several minutes. Let's see, there was something else in the chat. Do we have to do job submission to Midway, or can we use the Gardner cluster? I'm not sure what the Gardner cluster is. Whenever I've done this on Midway, I haven't actually submitted it as a dedicated job. I find that it is not that computationally intense. Sometimes it's more network intense, and it runs pretty quickly. So, so far I haven't had any of my downloads terminated on Midway for doing this. But if you find that to be the case, you could very easily just run the S Interactive tool, which will give you a dedicated Midway node that you can use to download this stuff to your Scratch space. Yeah, cool. Yeah, the CLI tool that Basespace does, you can download that on any Linux platform, and I'm pretty sure that it's compatible with all that stuff. All right, I don't want to take the time to go through this. I don't know how long it'll take to do reclustering, but this is basically just doing that last step of what's done with the Cell Ranger tool. Oh, there we go, it's done. And so here you can see now I've clustered out that secondary cluster here, which was a lot of dead cells were over there. But yeah, now you can do more analysis. I'm not sure how good this actual data set was. This isn't a data set that's sanctioned by 10x as a demo or anything. This is just some sequencing data that I got. But you can actually go in here and you can look at differentially expressed genes between all the different clusters. I don't think that this takes too long to run from what I remember. If you want to see the results of this, you can stick around. It should just take another couple seconds, I think. Are there any more questions while I'm waiting? Oh, here we go. Yeah, so it already popped up. So it gives you a list of upregulated genes per cluster. 10x, the Cell Ranger secondary analysis identified 7 different clusters of heterogeneity here. And so you can look at what are the genes that are largely upregulated significantly for each of those clusters. So here's, I guess, the June gene. I'm not sure what a lot of these genes are for, but yeah, so this is where you would start to do your secondary and tertiary analysis of your single cell sequencing data. But that's overall the pipeline of going from your sample that you loaded onto your sequencer and how you go from that to here. And this is my preferred workflow with using RCC to do this quickly. So that, again, if I happen to be traveling or something, or if I decide to be working at a coffee shop somewhere, I don't have to download everything onto my poor laptop using my phone's hotspot or something. You can just use the tools that are at UChicago, and I really like the 10x Cloud analysis tool rather than running Cell Ranger on my own computer. So yeah, that's what I have for you guys. If you have any more questions, feel free to put it in the chat. Otherwise, you can always email me. The CIIC always has tons of useful resources for this too. Yeah, my email is jono@uchicago.edu. It's just JONO, uchicago.edu. Yeah, oh, of course, and Emma's here. Yeah, so, and Heather's here. So yeah, they've been really helpful. Emma and Heather at 10x Genomics, they have a lot of very useful resources for dealing with us. So yeah, thanks everybody for your time, for attending this. Yeah, I believe that this is all recorded and will be uploaded, and I'll put these slides up somewhere too, which I think have a decent reference to some of these tools, maybe just links to places that have better references. What website will these be posted on? I imagine that it'll be the CIIC website. Fatih will probably send out an email about this. I'm not really sure what you want to do.
Yes. So thanks for joining us today. And thanks, Jonathan, for the great talk of handling sequencing data. We appreciate if you have any feedback and also have questions, feel free to send the email to Jonathan or send it to CIIC-singlecell@uchicago.edu. Lastly, we are looking for any speakers who would like to share the sequencing experience. Please reach out to us. Thanks for your time today.